Welcome to this comprehensive training on Analytical Quality by Design, or AQbD, and today we'll explore how this systematic approach is revolutionizing analytical method development in the pharmaceutical industry. By the end of this session, you will understand the fundamental principles of analytical quality by design, how AQbD differs from traditional analytical method development, the key components of the AQbD framework, practical implementation strategies, real-world case studies, and benefits. Traditional analytical method development has often been a trial-and-error process. Scientists would adjust one parameter at a time, hoping to achieve acceptable results. This approach is time-consuming, resource-intensive, and often fails to provide robust methods that perform consistently across different conditions. Analytical quality by design is a systematic approach to analytical method development that begins with predefined objectives and emphasizes product and process understanding through risk assessment and the systematic application of statistical tools. The concept builds upon quality by design principles established by the International Council for Harmonization, or ICH, particularly ICH Q8, Q9, and Q10 guidelines. AQbD is built on four fundamental principles. First, enhanced process understanding. We must thoroughly understand what we're trying to measure and why. This includes understanding the critical quality attributes of our analyte and the potential sources of variability. Second, risk-based approach. We systematically identify and assess risks that could impact method performance, then design experiments to mitigate these risks. Third, systematic experimentation. Instead of changing one variable at a time, we use designed experiments to efficiently explore the method design space and understand interactions between variables. Fourth, continuous improvement. AQbD methods are designed to be robust and include mechanisms for ongoing monitoring and improvement throughout their lifecycle. Traditional approach starts with literature review, followed by trial-and-error optimization, single-factor experiments, and finally, method validation. This often results in methods that work under specific conditions but fail when conditions change slightly. AQbD begins with defining the analytical target profile, conducting thorough risk assessment, using designed experiments to understand the method design space, and building in robustness from the start. The result is a method that performs consistently across its intended range of use. The AQbD framework consists of several interconnected elements. Let's examine each one in detail. The Analytical Target Profile is the foundation of AQbD. Think of it as a detailed specification of what the analytical method needs to achieve. The ATP includes purpose. What question is the method answering? Analyte. What are we measuring, including structural information? Matrix. What sample types will be analyzed? Range. What concentration range must be covered? Precision and accuracy requirements. How precise and accurate must the results be? Sample throughput. How many samples per day? Regulatory requirements. What guidelines must be met? Here's a practical example of an ATP for an HPLC assay. Develop a stability indicating HPLC method for quantitative determination of active pharmaceutical ingredient X in tablet formulations. The method must quantify the API from 80 to 120% of label claim with precision RSD, 0% in accuracy within 98 to 102%. It must separate API from known degradation products and excipients, with analysis time under 15 minutes per sample. Notice how specific and measurable each requirement is. Critical method parameters are those method variables that significantly impact the method's ability to meet its ATP requirements. These might include, for HPLC methods, mobile phase composition and pH, column temperature, flow rate, injection volume, detection wavelength. Risk assessment systematically identifies potential failure modes and their impact on method performance. We typically use tools like Ishikawa diagrams, fishbone diagrams, to identify potential sources of variability, risk matrices to assess probability and impact failure mode and effects analysis, FMEA, for comprehensive risk evaluation. The goal is to identify high-risk parameters that require careful control or further investigation. design of experiments is where AQbD really shows its power. Instead of the traditional one factor at a time approach, DOE allows us to study multiple factors simultaneously, identify interactions between variables, efficiently explore the design space, build predictive models. Common DOE designs include factorial designs, response surface methodology, and optimal designs. Let's look at a practical example. We're developing an HPLC method and have identified 3 critical parameters. Mobile phase pH, factor A, 2.5 to 3.5 acetonitrile percentage. Factor B, 35% to 45%. Column temperature, factor C, 25 degrees Celsius to 40 degrees Celsius. The method Operable Design Region represents the multi-dimensional space where the method consistently meets its ATP requirements. Think of it as the sweet spot where your method will work reliably. The MODR is defined by overlaying the acceptable ranges for all critical quality attributes. Operating within the MODR ensures consistent method performance. The control strategy defines how we'll ensure the method continues to meet its ATP throughout its life cycle. This includes system suitability tests, real-time checks that the method is performing acceptably, periodic verification, scheduled checks of method performance, change, control, procedures for managing method modifications, continuous monitoring, ongoing assessment of method performance trends. AQbD methods include built-in lifecycle management. This involves Stage 1, development, establishing ATP, conducting risk assessment, DOE studies. Stage 2, qualification, formal method validation within the MODR. Stage 3, continued verification, ongoing monitoring and improvement. Stage 4, method transfer, moving methods between laboratories. Stage 5, method retirement, formal decommissioning when no longer needed. Implementing AQbD in your organization requires careful planning. Here's a practical roadmap. Phase 1, education and training. Start by training your analytical team on AQbD principles. Everyone needs to understand the why behind the approach. Phase 2, pilot projects. Select 2, 3 simple method development projects as pilots. Choose methods where traditional approaches have struggled. Phase 3, tool implementation. Invest in statistical software for DOE and data analysis. Popular options include JMP, Design Expert, or Minitab. Organizations often face several challenges when implementing AQbD. Cultural resistance. Scientists comfortable with traditional approaches may resist change. Address this through education and demonstrating early wins. Resource investment. AQbD requires upfront investment in training and tools. However, this pays dividends through reduced method development time and fewer method failures. Regulatory concerns. Some worry that regulators won't accept AQbD methods. In reality, regulatory agencies encourage science-based approaches. Complexity. Perception. AQbD seems complex initially, but it actually simplifies method development once implemented. Success requires building organizational capability across multiple areas. Let's examine a real-world example. A pharmaceutical company needed to develop a stability indicating HPLC assay for a new drug product. Traditional approach results: 6 months development time, 3 method failures during validation, limited understanding of method robustness. AQbD approach. Results: 3 months development time, successful validation on first attempt, comprehensive understanding of method design space, robust performance across multiple laboratories. A biotechnology company used AQbD to develop an LC MS method for protein quantification. The systematic approach helped them identify critical sample preparation steps, optimize chromatographic conditions efficiently, understand method sensitivity to matrix effects, develop a control strategy ensuring consistent performance. Result: a method that performed consistently across different operators, instruments, and sample batches. 1. What are the 4 core principles of AQbD? 2. How does the Analytical Target Profile differ from traditional method specifications? 3. Name 3 tools commonly used in AQbD risk assessment. 4. What is the method Operable Design Region and why is it important? 5. List 3 quantified benefits organizations report from AQbD implementation.